Hello friends, today we will learn about antigen antibody reaction. So let's begin. Antigen In this video we will study about antigen antibody reactions, its general uses, characteristics, stages, measurements and types of antigen antibody reactions like precipitation reaction, agglutination, complement fixation test, Conglutination, neutralization test, opsonization, immunofluorescence, radioimmunoassay, RIA, enzyme-linked immunosorbent assay, ELISA, enzyme-linked immunosorbent assay, and chemiluminescence assay, immunoelectron microscopic test, immunoblotting. We'll discuss about this. Now let's begin with antigen antibody reaction. First, general feature. What are the features we will discuss in this now? What are the features? Antigen combines with its specific antibody in an observable manner, and the reaction between antigen and antibody is specific. These antigen antibody reactions in vitro are known as serological tests. Now let's know about the uses in the body or in vivo. It forms the basis of immunity against infectious diseases. It may lead to tissue injury in some hypersensitivity reactions and autoimmune diseases. Now in the laboratory or in vitro. For diagnosis of infections, helpful in epidemiological studies for identification of non-infectious agents such as enzymes, detection and quantitation of either antigens or antibody. Now let's know about the characteristics. Reactions is specific. An antigen combines with only with its homologous antibody and vice versa. However, cross reactions may occur due to antigenic similarity. Entire molecules of antigen and antibody reaction are not the fragments. Only the surface antigens participates in the antigen antibody reaction. The reaction is firm but reversible. The firmness of combination depends on the affinity and avidity. Affinity is defined as the intensity of attraction between antigen and antibody molecules. Avidity is the binding strength after the formation of antigen antibody complexes. Antigens and antibodies can combine in varying proportions. Antibodies are generally bivalent, although IgM may have 5 or more combining sites. Antigens may have valencies up to hundreds. Now let's know about the stages. Antigen antibody reactions occur in two stages. One is primary stage, another is secondary stage. Let's know about the primary stage. The initial interaction between antigen and antibody is rapid but without any visible effect. This reaction is reversible. The binding between the antigen and antibody occurs by the weaker intermolecular forces such as van der Waals forces, hydrogen bonds and ionic bonds rather than by farmer covalent binding. This primary interaction can be detected by estimating free and bound antigen or antibody separately in the reaction by a number of methods. Now let's know about secondary stage. The primary stage in most of the instances but not all is followed by secondary stage which has visible effects such as precipitation, agglutination, complement fixation, neutralization and immobilization of motile organisms. When these reactions were discovered, it was believed that different type of antibody was involved for each type of reaction. On this basis, the antibody involved in agglutination was called agglutinin and those involved in precipitation as precipitin and the corresponding antigens were called agglutinogen and precipitogen respectively. This concept was replaced by Ginzer's Unitarian hypothesis in 1920, which held that an antigen would induce synthesis of only one antibody, which produces different types of visible or observable effects. It is observe both of these extreme views are not true. It is now an established fact that a single antibody can cause different types of antigen antibody reactions, and a single antigen can stimulate production of different classes of immunoglobulins which differ in their reaction capacities as well as in other properties. Now let's know about the measurements. Measurement may be termed of mass. Measurement may be in terms of mass or more commonly as titer of units. The antibody titer is the highest dilution of the serum which shows an observable reaction in the particular antigen antibody reaction. Antigens may also be titrated against sera. Now let's know about the types of antigen antibody reactions. First is precipitation reaction. Let's know about the precipitation. When a soluble antigen reacts with its antibody in the presence of electrolytes, that means sodium chloride, NaCl, at an optimal temperature and pH, the antigen antibody complex forms an insoluble precipitate and it is called precipitation. The precipitate usually sediments at the bottom of the tube. Precipitation may occur in liquid media or in gels such as agar, agarose or polyacrylamide. Now let's know about flocculation. When instead of sedimenting, the precipitate is suspended as flocules. The reaction is called flocculation. This is a modified form of precipitation. A serological test, that is VDRL routinely used for the diagnosis of syphilis is one example of flocculation test. Now let's know about Proson phenomenon. When same amount of antisera is mixed with increasing quantities of antigen is in different tubes, the precipitation takes place in one of the middle tubes in which antigen and antibody are present in optimal or equivalent proportion. In the preceding tubes, antibody is in excess, and in latter tubes in which antigen is in excess, the precipitation is weak or even absent. The absence of precipitation in the presence of excess antibodies is known as Proson phenomenon. This is really valent clinically, as this sera should be diluted further to get the proper results of precipitation. Now let's know about mechanism of precipitation. A scientist called Marak in 1934 proposed the lattice hypothesis, which is widely accepted. According to this concept, multivalent antigens combined with bivalent antigens, precipitation occurs when only only a large lattice is formed. This occurs in the zone of equivalence. In the zone of antigen access, in the zone of antigen excess, the valency of the antibody are fully satisfied, which results in the failure or to form a large lattice. Similarly, in the zone of antibody excess, the valencies of the antigen are taken up with antibody and the result in failure is to form a large lattice. This lattice hypothesis holds true for agglutination also. Now let's know about the application of precipitation reaction. The precipitation reaction may be used either as qualitative or quantitative test. It is very sensitive test and can detect as little as 1 microgram of protein antigen. Precipitation test has a considerable value in some points. Let's know about these points. First is identification of bacteria, example Landsfield's groups of streptococcus. Second point, detection of antibody for diagnostic purposes, example VDRL in syphilis. Third point, forensic application in identification of human blood and seminal stains. Fourth is testing for food adult adulterants. Fifth point, to standardize toxins and antitoxins. Now let's know about the types of precipitation reaction. First is ring test. Antigen solution is layered over the antiserum in a narrow tube. A precipitate ring appears at the junction of the two liquids. Examples of ring test are C-reactive protein, in short CRP test, and streptococcal grouping by the Landsfield technique. Second point is flocculation test. Second test is flocculation test. It can be done in the slide and in the test tube. Now let's know about the slide test. A drop of antigen solution is added to a drop of inactivated patient's serum on a slide and mixed by shaking. Floccules appear. The VDRL test for detection of antibodies against syphilis is an example of slide flocculation test. Now let's know about the tube test. The Kahn test, that means tube flocculation, was done previously for diagnosis of syphilis. Standardization of toxins and toxoids is also done by tube flocculation test. Now let's know about immunodiffusion test. These are precipitation tests in gel. Immunodiffusion is generally done in 1% agar gel. There are following advantages of precipitation in gel rather than in a liquid medium. The reacting band is easily visible and can be stained for preservation. This band is stable. The number of different antigens in the reaction can be observed as each antigen antibody reaction gives rise to a line of precipitation. Therefore, it helps to identify different antigens. Identity cross reaction and identity between different antigen can also be observed by immunodiffusion. Now let's know about the various types of immunodiffusion test. First is single diffusion in one dimension. It can be called as Woodin procedure. The antibody is mixed in agar gel in a test tube and antigen solution is poured over it. The antigen diffuses downward through the agar gel and a line of precipitation is formed. The number of precipitate bands will indicate the number of different antigens present. It is single diffusion of antigen only and in one dimension, that is towards antibody in agar gel. Second point, we will know about double diffusion in one dimension. In short, it is otherwise known as Ukle-Falthrop procedure. The antibody is incorporated in agar gel and this above gel, which is placed in a column of a plain agar. The antigen is poured on top of this plain agar. The antigen and antibody move towards each other through intervening column of plain agar and a precipitate band is formed where they meet in optimum concentration. Now let's know about single diffusion in two dimensions. In short, it is otherwise known as radial immunodiffusion. The antiserum, and that means antibody, is incorporated in agar gel on a slide or petri dish. The wells are caught on the surface of gel. Antigen is added to the wells. The antigen diffuses radially and ring-shaped band of precipitation are formed around the well. The diameter of the ring is directly proportional to the concentration of the antigen. Now let's know about the uses. It has been widely employed for estimation of immunoglobulin classes, example IgG, IgM, IgA in sera. Next point is it has also been used for screening sera for antibody to influenza viruses. Now let's know about double diffusion in two dimensions. It is otherwise known as Uchter's-Roni procedure. Agar gel is poured on a slide. Wells are caught using a template. The antiserum or antibody is placed in the central well and surrounding wells are filled with the different antigens. If two adjacent antigens are identical, the lines of precipitate formed by them will fuse with each other. If they are unrelated, the lines cross each other. If they are unrelated, the lines cross each other. Spore formation indicates each reaction or partial identity. A special variety of double diffusion in two dimensions is the ELEX test for toxigenicity in C. diphtheria. Now let's know about immunoelectrophoresis. Immunoelectrophoresis combines electrophoresis and immunodiffusion. This is done on a glass slide layer with semi-solid agar. A well, a well is cut and antigen is filled. The first step is electrophoresis of antigen for about 1 hour. Rectangular trough is caught in the agar parallel to the direction of migration of antigen and filled with antibody. Diffusion is allowed to proceed for 18 to 24 hours. Precipitation lines develop with each separated component of the antigen. By this technique, a number of antigens can be identified in human serum. It is particularly useful for detection of normal and abnormal serum proteins like myeloma proteins. Now let's know about electroimmunodiffusion. Immunodiffusion can be speeded up if antigen and antibody are driven by electricity. It is combination of electrophoresis and diffusion. Of these methods, counter immunoelectrophoresis and rocket electrophoresis are used frequently. Now let's know about counter immunoelectrophoresis. It is one-dimensional double electrophoresis immunodiffusion. This test is based on the movement of antigen and antibody in opposite direction. This is performed on a glass slide layered with agarose and wells are caught on the surface. One well is filled with antigen and other with antibody. Electric current is passed through the gel. The antigen and antibody move towards each other, resulting in a participation line at a point between them. It takes about 30 minutes and is more sensitive than standard immunodiffusion technique. It is applied clinically for detecting hepatitis B antigens and antibodies. Antigens of cryptococcus in cerebrospinal fluid and in number of other diseases. Now let's know about rocket electrophoresis. It is one-dimensional single electroimmunodiffusion. This technique is mainly applied for quantitation of antigens. The antiserum to the antigen to be quantitated is mixed in agarose and gelled on a glass slide. The wells are punched in the set gel and filled with increasing concentrations of the antigen. It is then electrophorused. Participation is formed in the shape of cone-like structures. The length of these rocket-like structures corresponds to the concentration of antigen. Now let's know about third point, that is L'Oreal two-dimensional electrophoresis. It is a variant of rocket electrophoresis. By this method, several antigens in a mixture can be quantitated. In the first stage of the technique, the antigen mixture is electrophoretically separated, and in the second stage, electrophoresis is done perpendicular to that of first stage to get rocket-like precipitation. Now let's know about agglutination. It is an antigen-antibody reaction in which a particular antigen combines with its antibody in the presence of electrolysis lights at an optimal temperature and pH, resulting in visible clumping of particles. It differs from precipitation, in which soluble antigen is present in contrast to particulate antigen of agglutination. The agglutination is more sensitive than precipitation for the detection of antibodies. The agglutination reaction takes place better with IgM antibody than with IgG antibody. Principles governing agglutination are the same as that of the precipitation. Agglutination occurs when antigen and antibody are present in optimal proportions. Lattice formation hypothesis holds good for agglutination too. The zone phenomenon may occur when either an antigen or an antibody is in excess. Occasionally, incomplete antibodies are formed that combine with the antigen but do not cause agglutination. These act as blocking antibodies, inhibiting the agglutination by the complete antibody added subsequently. Now, let's know about the types of agglutination. First is slide agglutination test. A uniform suspension of antigen is made in a drop of saline on a slide or tile, and a drop of appropriate antiserum is added. The agglutination reaction is facilitated by mixing the antigen and the antiserum with a wear loop or by gentle rocking the slide. Clumping occurs instantly or within seconds when agglutination test is positive. Clumping after a minute may be due to drying of the fluid and should be disregarded. A control consisting of antigen suspension in saline without adding antiserum must be included on the same slide. It is to ensure that antigen is not auto-agglutinable. Now let's know about uses. It is a routine procedure to identify the bacterial strains isolated from clinical specimens. One example is to identify Salmonella species. It is also used for blood grouping and cross matching. Now let's know about tube agglutination test. This is a standard quantitative method for determination of antibodies. Serum is diluted serially by doubling dilution in test tubes. An equal volume of a particulate antigen is added to all tubes. The highest dilution of serum at which agglutination occurs is antibody titer. Tube agglutination is routinely employed for antibody detection in diagnosis of typhoid, brucellosis, and typhus fevers. Now let's know about the uses of tube agglutination test. First is serological diagnosis of enteric fever, typhus fever, infectious mononucleosis, brucellosis. For diagnosis of primary atypical phenomenon, pneumonia. Atypical pneumonia. Now let's know about complications related to tube agglutination. Two main complications may be: first is Projon phenomenon, second is blocking antibody. Let's know about the Projon phenomenon. Serum with high concentration of antibodies may be not able to react with antigen and gives a false negative result. Several dilutions of the serum should be tested to prevent such false negative results due to frozen. Now let's know about the blocking antibody. Blocking or incomplete antibodies may be detected by performing the test in hypotonic saline or albumin saline. Antiglobin test is more reliable for detecting these antibodies. Now let's know about the antiglobulin test, or otherwise known as Combs test. This test was devised originally by Combs, Morant and Race in 1945 for the detection of incomplete anti-RH antibodies. When sera containing incomplete antibodies Rh positive and Rh positive erythrocytes, the incomplete antibody globulin coats the erythrocytes but no agglutination occurs. When such coated erythrocytes are treated with antiserum, with antiglobulin or comb serum, that is from rabbit antiserum against human uh, gamma globulin, the cells are agglutinated. This principle is called Combs test. There are two types of Combs test. First is direct test, another is indirect Combs test. First is direct Combs test, second is indirect Combs test. Let's know about direct test. This the only difference between two is that the sensitization of the erythrocytes with incomplete antibodies takes place in vivo in direct type, whereas it occurs in vitro in indirect type. Now let's know about the uses of Combs test. For the detection of anti-RH bodies, first second is for demonstration of any type of incomplete antibody, example brucellosis. Now let's know about heterophile agglutination test. Heterophile antibodies has a property to react with microorganisms or cells of unrelated species due to common antigenic sharing. Now let's know about well-Felix reaction. Some protease stains are agglutinated by sera of patients with rickettsial infections. This is due to antigenic sharing between these protease stains and rickettsial species. Now let's know about Paul Bunnet test. Sheep erythrocytes are agglutinated by sera of infectious mononucleosis. And next third test is Streptococcus mg agglutination test. It is positive in primary atypical pneumonia. Now let's know about the passive agglutination test. A precipitation reaction can be converted into agglutination test by attaching soluble antigen to the surface of carrier particles such as latex particles, bentonite and red blood cells. Such tests are called passive agglutination test. This conversion is done because agglutination tests are more sensitive for detection of antibodies. Passive agglutination tests are very sensitive when instead of antigen, the antibody is absorbed on the carrier particles. For estimation of antigens, it is known as reversed passive agglutination. Now let's know about latex agglutination test. Polystyrene latex particles are widely employed to adsorb several types of antigens. Latex particles can also be coated with antibody for detection of antigen. These tests are very covalent, very convenient, rapid and specific. These are used for detection of hepatitis B antigen, ASO, CRP, RA factor, HCG, bacterial typing and many more antigens. Now let's know about hemagglutination test. Erythrocytes sensitized with antigen are used for detection of antibodies in rheumatoid arthritis. An antibody, that is RA factor, appears in the serum. This RA factor acts as an antibody to gamma globulin. Thus, RA factor can agglutinate red cells coated with gamma globulins. The antigen used for the test is CIP red blood cells sensitized with rabbit anti-CIP erythrocyte antibody. The amboceptor is antibody, therefore gamma globulin in nature. This is the principle of Rose-Waller test employed for detection of RA factor in cases of rheumatoid arthritis. Now let's know about coagglutination. First is principle. It is based on a presence of protein A on the surface of some strains of Staph aureus. Specific IgG immunoglobulin is coated on this COAN1 strain of Staphylococcus aureus. Fc portion of IgG molecule binds to protein A, whereas antigen combining FAB terminal remains free. When the corresponding antigen is mixed with these coated cells, FAB terminal binds to antigen resulting in agglutination. This test is used for detection of bacterial antigens in blood, urine and CSF. N. gonorrhea, streptococcus pyogenes and H. influenza antigens can be detected by this method. Now let's know about complement fixation test. In short, CFT. Principle. The antigen antibody complexes have ability to fix complement. This reaction has no visible effect. To detect the fixation of complement, an indicator system consisting of CIP erythrocytes coated with amboceptor is used. Complement can be canalized. These erythrocytes coated with antibodies. If complement is fixed and utilized in the antigen-antibody reaction, there is no fire complement to act on the indicator system and hence no lysis of erythrocytes, which indicates the positive complement fixation test. Lysis of erythrocytes indicates that complement was not fixed in the first step and therefore the serum is negative for antibodies. Now let's know about procedure. The serum should be inactivated by heating and 56 degrees centigrade for 30 minutes to destroy any complement activity and activity of the serum may have and to remove some non-specific inhibitors of complement. The antigen may be used as soluble or particulate. Fresh guinea pig serum is used as source of complement. Complement activity is heat liable, so it is used as fresh. Now let's know about standardization of complement. The guinea pig serum is titrated for complement activity. 1 unit of minimum hemolytic dosage, that is MHD, of complement is defined as the highest dilution, that is smallest quantity of guinea pig serum which lyses 1 unit volume of washed CIP red blood cells in the presence of excess hemolysin within a fixed time, that is 30 to 60 minutes, and at a fixed temperature, that is 37 degrees centigrade. Now let's know about the titration of amboceptor. The amboceptor is titrated for hemolysin activity. 1 unit of minimum hemolytic dosage of hemolysin is defined as the highest dilution, that is smallest quantity of the inactivated amboceptor which lyses 1 unit volume of washed CIP red blood cells in the presence of excess complement within a fixed time and at a fixed temperature. Normal saline with added calcium and magnesium is used as diluent for titrations of complement amboceptor and for CFT. Now let's know about the indirect complement fixation test. Certain avian Example, dog, parrot and mammalian. Example, horse, cat, Sarah cannot fix, guinea pig, complement. Indirect complement fixation test is used for testing such Sarah. Test is done in duplicate and after the first step. The standard antiserum known to fixed complement is added in one set. First step, that is antigen plus test serum plus guinea pig complement. Second step is standard antiserum will react with antigen and fix the free complement. Indicator system. Let's know about the indicator system. That means no hemolysis because complement is not free to act on indicator system. In a positive test, antigen would have been used up by serum in the first step and standard antiserum would not be able to fix the complement which remains free to act on indicator system resulting in hemolysis. First step is antigen plus test serum plus guinea pig complement. Second step is standard antiserum cannot react with the antigen because antigen has been used up by antibody in the first step but complement is free as it is not fixed. Now let's know about indicator system. Indicator system here is hemolysis occurs because complement is free to act on the system. Now coagulation. Conglutination. Let's know about the conglutination. This is an alternative method for systems which do not fix guinea pig complement. Horse complement is used. The indicator system is sheep erythrocyte sensitized with bovine serum. Bovine serum contains a beta globulin component named conglutenin, which acts as antibody to complement. Conglutenin can cause agglutination of sensitized sheep erythrocytes if these are combined with complement. This process is called conglutination. First step here is antigen plus antiserum, that is positive, and horse complement gives rise to complement is fixed. Second step is sheep erythrocytes with conglutenin. No agglutination occurs because complement is not free to act with sensitized erythrocytes. No agglutination, that is positive result. Agglutination, negative result. Let's know about the other complement dependent serological test. First is immobilization test. When live motile Treponema pallidum is mixed with patient serum in the presence of complement, the organism becomes non-motile. This is the principle of Treponema pallidum immobilization test. Next is immune adherence. The antigen antibody complexes is in some bacteria like V. cholera, T. pallidum adhere to particulate material such as erythrocytes or platelets. This bacterial adherence to cells is known as immune adherence. Now let's know about cytolytic or cytocidal test. When a live Vibrio cholera is mixed with its antibody in the presence of complement, the bacterium is lysed. This forms the basis of measurement of anti-cholera antibodies. Now let's know about neutralization test. Bacterial exotoxins are capable of producing neutralizing antibodies which play a role in protection against diseases such as diphtheria and tetanus. The toxicity of bacterial endotoxins is not neutralized by their antibodies and these are named as virus neutralization test. Toxins and antitoxin neutralization can be measured in vivo and in vitro. Now let's know about in vivo test. First test is toxigenicity test. Toxigenicity test is done for detection of toxin of C. diphtheria. It is an intradermal test to inject bacterial toxin in animal previously protected by antitoxin of C. diphtheria and a test animal without antitoxin. No biological effects of toxin are observed in the control animal, but test animal dies. Now let's know about seek test. This is similar kind of test in humans. Diphtheria toxin is injected intradermally in man. There is no reaction at the site of injection if person is immune to diphtheria, that is antitoxin is present in blood. Injected toxin is neutralized by circulating antitoxin. Now let's know about in vitro test. First test is anti-streptolysin O test. In short, ASO test. Serum of patients suffering from Streptococcus pyogenes infection contains anti-streptolysin O, which neutralizes the hemolytic activity of streptococcal O hemolysin. Now let's know about virus neutralization test. Neutralization of viruses can be demonstrated in cell cultures, eggs and animals. It is used mostly in typing viral isolates. Now let's know about Nagler reactions. Clostridium welchii toxin is neutralized by antitoxin when the bacteria is grown in egg yolk medium containing antitoxin. It is useful for rapid detection of C. welchii in clinical specimens. Now let's know about opsonization. Opsonization is the process by which a particulate antigen becomes more susceptible to phagocytosis. This occurs by help of opsonin, which combines with an antigen and facilitates phagocytosis. Opsonin may be an antibody-like substances or other component present in serum. Opsonic index is defined as ratio of phagocytic activity of the patient's blood for a given bacterium to that of a normal individual. Phagocytic index is the average number of phagocytosed bacteria per polymorphonuclear leukocyte from stained blood films. Phagocytic index denotes the phagocytic activity of the blood and thus help in measuring opsonic index. Now let's know about immunofluorescence. Fluorescence is the property of certain dyes which absorb rays of one particular wavelength and emits rays with a different wavelength. Cones and his colleagues in 1942 showed that fluorescent dyes can be conjugated to antibodies, and these labeled antibodies can be used to detect antigens in tissues. The commonly used fluorescent dyes are fluorescein, isothiocyanate and lysamine rhodamine, exhibiting blue-green and orange-red fluorescent respectively. Immunofluorescent test is of two types. First is direct immunofluorescent test and indirect immunofluorescent test. Let's know about the direct immunofluorescent test. First is principle. Let's know about the principle. The specific antibodies tagged with fluorescent dye are used for detection of unknown antigen in a specimen. If antigen is present, it reacts with labeled antibodies, and fluorescence can be observed under ultraviolet light of fluorescent microscope. This is depicted and let's know about the useful uses. It is commonly employed for detection of bacteria, viruses or other antigens in blood, CSF, urine, feces, tissues and other specimens. It is sensitive method to diagnose rabies by detection of rabies virus antigens in the brain smear. Now let's know about the disadvantage. Separate specific fluorescent labeled antibody has to be prepared against each antigen to be tested. Now let's know about indirect immunofluorescence test. The indirect method is employed for detection of antibodies in serum or other body fluids. Now let's know about the principle. A known antigen is fixed on a slide. The known antibody is applied to the slide. If antibody is present in the serum, it attaches to known antigen on the slide. For detection of this antigen-antibody reaction, fluorescence tagged antibody to human globulin is added. In positive test, fluorescence occurs under ultraviolet light. One specific example of detecting antibodies in serum of syphilis patient is also included. Now let's know about the advantages. A single anti-human globulin fluorescent conjugate can be employed for detection of antibody to any antigen. All antibodies are globulin in nature, therefore anti-human globulin attaches to all antibodies. This has overcome the disadvantage of direct immunofluorescence test. Now let's know about sandwich techniques of immunofluorescence. Antigen being in the middle with labeled and unlabeled antibody on either side forms a sandwich. This is used for detection of antibodies. Antibody plus antigen plus labeled antibody. Now let's know about RIA, that is radioimmunoassay. Burson and Allo in 1959 first described the test radioimmunoassay, and since then it has been utilized for quantitation of hormones, drugs, hepatitis B surface antigen, IgG, and viral antigens. In 1977, Yallo was awarded the Nobel Prize for the discovery of RIA test. This test can detect antigens up to picogram, that means 10 to the power minus 12 gram quantities. RIA is based on a competition of a fixed amount of specific antibody between known radio-labeled antigen and unknown unlabeled antigen. The competition is determined by the level of the test antigen present in the reacting system. After antigen antibody reaction, the antigen is separated into the free and bound fraction, and their radioactivity is measured. The concentration of test antigens is calculated from the ratio of the bound and total antigen labeled using a reference curve. Now let's know about the ELISA, enzyme linked immunosorbent assay. ELISA has been applied widely for the detection of a variety of antibodies and antigens. It is simple and nearly as sensitive as radioimmunoassay. It requires only microlite quantities of test reagents. The principle of ELISA is almost same as that of immunofluorescence. The only difference being an enzyme is used instead of fluorescent dye. The enzyme acts on substrate to produce a color in a positive test. ELISA can be used for detection of antigen or antibody. Tests for specific immunoglobulin classes are also available. It is done on a solid phase. The test can be done in polystyrene tubes or polyvinyl micro titer plates. Now let's know about sandwich ELISA. For antigen detection in a specimen, the walls of microtiter plates are coated with specific antibody against the antigen to be detected. Specimens to be tested are added in coated walls. If antigen is present in specimen, it binds to coated antibody. To detect this antigen-antibody reaction, antiserum conjugated with an enzyme is added. This conjugated antiserum binds to an antigen already attached to coated antibody. A substrate is added to know the binding of conjugated antiserum of antigen antibody complex. In case of binding, an enzyme acts on substrate to produce color, intensity of which can be read by spectrophotometer or ELISA reader. Color detection can also be seen by naked eye. This type of ELISA test is also known as sandwich ELISA. Positive and negative controls should always be included in the test. At every step of ELISA test, incubation and washing is done to wash up unbound reagents. Now let's know about indirect ELISA. For the antibody detection, the wells of microtiter plate are coated with antigen. Sera to be tested are added in these coated wells. If antibody is present in specimen, it binds to coated antigen. To detect this antigen-antibody reaction, a goat anti-human immunoglobulin antibody conjugated with an enzyme is added. Enzyme conjugated anti-human immunoglobulin binds to antibody, and to detect this binding, a substrate is added. An enzyme acts on substrate to produce color in a positive reaction. This procedure is also named as indirect ELISA. Now let's know about the competitive ELISA. It has been used for detection of HIV antibodies. Positive result shows no color, whereas appearance of color indicates a negative test. Like radioimmunoassay, there are two specific antibodies, one conjugated with enzyme and other present in serum. Competition occurs between two antibodies for same antigen. The microtiter plate wells are coated with HIV antigen. Sera to be tested is added to this wells and incubated at 37 degrees centigrade and then washed. If antibodies are present, antigen-antibody reaction occurs. To detect this reaction, enzyme-labeled specific HIV antibodies are added. There is no antigen left for these antibodies to act. These antibodies remain free and washed off during washing. Substrate is added, but there is no enzyme to act on it. Therefore, positive results show no color. If serum to be tested is negative for antibodies, antigen is there to combine with enzyme conjugated antibodies, and enzyme acts on substrate to produce color. Now let's know about cassette or cylinder ELISA. It is a simple modification of enzyme for simple modification of ELISA for testing one or few samples of sera at a time. The test is rapid as compared with the 2 to 4 hours taken for micro ELISA. The result is read visually. Cassette ELISA is being used for detection of HIV type 1 and type 2 antibodies. Specific type 1 and 2 antigens are immobilized on the nitrocellulose membrane in the cassette. Test serum is added on the membrane. In positive serum, antibody will bind to appropriate antigens. After washing, to remove unbound antibody, conjugate the enzyme labeled anti-human immunoglobulin antibody is added. It is washed again to remove unbound conjugate, and a substrate is added. A positive result shows a colored spot. Human immunoglobulin immobilized at a spot on the membrane serves as a control, as shown in development of colored at that site. Now let's know about the use of ELISA. How ELISA is used? It has been used for detection of antigen and antibodies of various microorganisms. Let's know about some examples. First is detection of HIV antibodies in serum. Second is detection of mycobacterial antibodies in tuberculosis. Third is detection of rotavirus in fasciaceae. Detection of hepatitis B markers in the serum. Fifth point is detection of enterotoxin of Escherichia coli in fasces. Now let's know about chemiluminescence assay. Chemiluminescence refers to a chemical reaction in which energy is emitted in the form of light. On the basis of this property, chemiluminescence compounds are used in this method to provide the signal during the antigen antibody reaction. The emitted light can be measured and the concentration of the analyte calculated. The method has been fully automated. This property of luminescence has also been applied for drug sensitivity testing of M. tuberculosis. Now let's know about immunoelectron microscopic test. First point is immunoferritin test. Ferritin, that is electron dense substance conjugated antibody, is used to react with an antigen. This antigen antibody reaction can be visualized under electron microscope. Second point is immunoelectron microscopy. This is in viral particles are mixed with specific antisera and are observed under the electron microscope. These are seen as clumps. This method is applied in some viruses such as hepatitis A virus and viruses causing diarrhea. Now let's know about immunoenzyme test. Some enzymes such as peroxidase can be conjugated with antibodies. Tissue sections are treated with peroxidase labeled antisera to detect corresponding antigen. The peroxidase bound to the antigen is visualized under the electron microscope. Now let's know about immunoblotting. In immunoblots, antibodies can detect proteins in mixtures. The mixture of proteins is electrophoretically separated in a gel. The separated proteins are then transferred from gel to a nitrocellulose paper. These nitrocellulose paper strips are reacted with test sera and subsequently with enzyme conjugated anti-human immunoglobulin. A suitable substrate is added. Color is produced by enzyme where specific antibody in test sera has reacted with separate protein in on strip. This test has been widely used to confirm the ELISA positive HIV antibody cases. This is known as Western blot test. It detects antibodies against various proteins fractions in test sera. Finally finished. Any questions regarding this, you can ask me in the comment box and subscribe my channel to know more updates. Thank you.